All right, we're going to get things kicked off here. So first and foremost, Happy New Year, everyone. And thanks once again for joining us today. And welcome to the 7th episode of Ewings U. I'm Piero Spada, and once again, I'll be your host again today. I also happen to be one of the co-founders of the Little Warrior Foundation and dad to 12-year-old little warrior Lucy. Today, we're excited to be hearing from Dr. Claudia Rössig to provide us all with some hope on an exciting and upcoming immunotherapy clinical trial. Briefly, Dr. Claudia Rössig received her medical degree at the University of Lübeck in Germany, then joined the Department of Peds Hem-Onc at University Children's Hospital in Munster, Germany, as a clinical fellow. Between 1998 and 2000, she was a postdoctoral fellow with Malcolm Brenner in the Center for Cell and Gene Therapy at Baylor College of Medicine in Houston, Texas, in the United States. After finishing her clinical training as a pediatrician in 2005 and her specialty registration as a pediatric hem-onc doctor in 2007, she is now director of the Department of Pediatric Hematology and Oncology in Munster, Germany. Her experimental research focuses on development of cellular immune therapeutic strategies to treat pediatric malignancies, including both leukemias as well as solid tumors. And as we'll see in a moment, she's a principal investigator of an innovative CAR-T clinical trial for pediatric solid tumors looking at very specific antigens, otherwise known as biomarkers. But before we kick things off with Dr. Rössig, I just want to let everybody know and remind you all that Ewings U is a webinar and podcast production of the Little Warrior Foundation. We are a childhood cancer foundation with a laser focus on finding a complete and permanent cure for Ewing sarcoma. We were founded in April of 2020 and have granted out $1.4 million in high potential therapies for increasing survivorship and decreasing toxicity. We very much so rely on the support of the communities of big and little warriors who are joining us on this webinar today. We are really appreciative of all of our communities' support to continue our mission so that we can continue to bulldoze our way towards a solution and continue this vital work of ours. With that, I'm going to hand it off to Emily to introduce herself and touch on a couple of housekeeping items.
Hi, I'm Emily McFadden, one of the other co-founders of Little Warrior Foundation. Thank you for joining us today. I'd like to call your attention to the bottom of your Zoom screen where there is a Q&A button. Throughout Dr. Rössig's presentation, please feel free to submit your questions there. Piero and I will be reading through those questions, and at the conclusion of her remarks, we will present some of those questions to her. As always, we cannot discuss any specific cases or provide specific medical advice during this webinar. If you'd like to see past versions of Ewings U, you can find them on our blog at littlewarrior.org. This session is also being recorded and will be available. The recording will be available on the blog in the coming week. With that, I will pass it over to Dr. Rössig.
Hello, everyone. It's my pleasure to give you an update on immunotherapeutic approaches for Ewing sarcoma. And while I'm sharing my screen, yeah, here we go. So I promised I'll give you an update on our planned clinical study, which will include Ewing sarcoma patients, but I'll also start from scratch, just giving you an insight into immunotherapeutic approaches to cancer in general, and also give you some explanations why so far nothing has really worked so well in Ewing sarcoma and how we could potentially change this in the future. So this is an illustration how well T cells are able to control cancer under the right circumstances. So what you see here is a patient with a disseminated cancer. I'm telling you it's not Ewing sarcoma, but I'm still showing you this image, this example on purpose. This is a patient I'm not sure you're all familiar with this type of imaging. What you're supposed to see is the heart, the kidneys, and the bladder, and also the brain a little bit, and everything else here is tumor manifestation. This patient was treated not with chemotherapy or anything else, just with 4 doses of immunotherapy, an oral, not oral, an IV agent, giving once every 3 weeks. And after just 4 doses, the cancer had entirely appeared. And actually, this was a 14-year-old girl. She has remained in remission ever since, and a couple of years ago. So I'm telling you, it's not Ewing sarcoma, it's a Hodgkin lymphoma. I was still presenting this example because it illustrates the power of T cells in general to even eliminate disease and maintain the remissions for a long time. So this is our goal also for other still incurable cancers. So what happened in this patient? How do these checkpoint inhibitors work? Tumors are not just heaps of malignant cells, masses of cells. They are actually kind of organs that consist of various individual cells, and some of these cells are immune effector cells. And if you liberate these immune effector cells, they can become active and attack the tumor cells and do what the immune system is good for, protect the body against life-threatening challenges. And the way this is achieved is that typically these T cells, these immune effector cells, when they enter a tumor, they become immediately tolerant and exhausted. And that's why the tumor cells, that's because the tumor cells upregulate signals that tell these T cells to stay away and not attack them and to just go to sleep. And these checkpoint inhibitors are agents that interrupt this communication. So the T cells wake up and realize there is a life-threatening cancer, and then they attack the cancer cells and can eliminate even the last cell. So that is how it should work. So far, this has worked in quite a substantial number of cancers, but unfortunately, all adult cancers, not pediatric cancers, but these checkpoint inhibitors really have made an impact in the past years and have turned incurable cancers, like, for example, certain types of lung cancer, into chronic diseases or even cures. And this has led to the Nobel Prize in Medicine 2018. So here is the frustrating slides. It shows responses in sarcomas, including Ewing sarcomas. Ewing sarcomas are the pink ones. And this is the kind of graph where all columns going upward are patients who did not respond at all. And only the patients here, these 3 patients, showed some minor responses. And there was only one Ewing sarcoma that showed at least a little bit of tumor shrinkage, but still not up to 30% reduction. So this is still disappointing. And all the other patients, including all these Ewing sarcoma patients, did not respond at all. This was just one study of several, because when the checkpoint inhibitor story came up, of course, there were lots of also pediatric cancer studies that tested whether checkpoint inhibitors could make a difference, and it couldn't. So why is that? It has been understood in the meantime that in order for the checkpoint inhibitors to work, there have to be several prerequisites have to be fulfilled. And one is that in the tumor microenvironment, T cells are actually present, and these must be T cells that have the inherent capacity to recognize cancer cells as foreign. And so checkpoint inhibitors have a high chance of working in a hot, so-called, we call it an inflamed or hot tumor, where you have lots of T cells. They're incapacitated by the tumor, but when you come up with the right agents, they wake up and eliminate the tumor. Some cancers are able to exclude the T cells from the tumor microenvironment. That is also a situation where checkpoint inhibitors don't work, but it might be possible to combine them with other agents that make these T cells infiltrate the tumors. And there is this completely frustrating situation also, where the tumors are infiltrated by all kinds of cells, but not by T cells at all. And so if there are no T cells, you cannot make the T cells work. It's obvious. But that is how this story ended, at least in the case of Ewing sarcoma and most other pediatric cancers. There's nothing to be revived, so checkpoint inhibitors won't do the trick. And what this made people think about was introducing T cells into the tumors. So if there are no T cells that recognize tumors, why not enable T cells to recognize tumors and then give them to the patient? along with checkpoint inhibitors or on their own. And what I described here is so-called adoptive T-cell therapy. The idea has been followed quite several decades ago. It's not such a new idea, but even in the 70s, 80s, people started thinking about T cells as magic bullets because their capacity to eliminate tumor cells has been recognized. But only recently, a technique has been invented that has enabled us to redirect T cells toward recognition of tumor-associated antigens. Even this idea is not so young. This is Zelik Eshar, who works at Weizmann Institute in Israel. And he has come up in the late 80s. He's an immunologist, and he was just studying T-cell receptors, so antigen recognition domains by T cells, finding out in which way T cells recognize their target. And so he has studied these receptors that enable T cells to engage with target antigen. And by doing this, he has come up with the idea of combining antibody recognition domains with T cell receptor components. And so he has produced these chimeras, where normally T-cell recognition is a process that involves a lot of individual steps and has a lot of requirements, especially for the presentation of antigen, whereas antibodies simply recognize antigen presented on the surface of the cell without any further requirements. And by combining these two technologies or these two biological means into one cell, he was able to engineer T cells that recognize surface-expressed antigens. And this has not really been his plan. He's an immunologist. He's not an immune therapist. He wasn't thinking of targeting cancer, but it has been picked up by researchers who thought this could be an ideal treatment for patients with incurable cancers. This has been successfully developed first for patients with leukemias and lymphomas. And here is really a wonderful success story of the field of pediatric oncology, a disease, one of the most frequent diseases we are targeting in pediatric oncology, acute lymphoblastic leukemia. We can cure a lot of patients with this disease, but certainly not all. So the cure rates are in the range of 80%. And this child here, Emily Whitehead, was one of the patients who failed all previous therapies. So in those cases, the disease becomes incurable. And she was treated with a single dose of chimeric receptor engineered T cells. And I show you in more detail what this means to this child and to future children. And she survived this cancer, this incurable cancer. She's posting a picture every year, and we were all waiting for her 10 years picture. I think it was in May when we saw that Emily has survived 10 years after salvage treatment with the CAR T cells, which probably means cure of the leukemia. And she's not the only patient who survived. This is illustrating the first clinical study performed in leukemias, in pediatric leukemias. And you can see that in a situation where typically the slope would go down to 0%, all incurable cancers, we now have a plateau of event-free, relapse-free survival in the range of 40%. And this has been now reproduced by many investigators and clinicians in many areas of the world. Real-world experience also always comes up with this around 40% cure rates in a previously incurable cancer. So that's not the ultimate solution yet. There's always still 60% of patients, even with this cancer, still failing treatment, but it's a major achievement. It's what we want to translate now to solid cancers. So CAR T-cell therapy, how does it work? What does it mean for the patient? First of all, you need to collect T cells because it's the patient's T cells that are gene modified to recognize the cancer. So you have to collect these T cells, and you do that from peripheral blood. The patient is connected to a machine for around 3 hours, 4 hours. It doesn't hurt, but you need a central line, and typically a normal Port or Hickman catheter is not sufficient, so you have to put in an additional central line under general anesthesia. And then the patient is connected to this machine. The T cells are collected and put into a bag and transferred to the lab. And then it takes a couple of days or weeks. This treatment in the leukemia patients, today, the pharmaceutically approved agents take several weeks still, but there are also procedures now, and this includes the technology we are developing for Ewing sarcoma patients, only takes around a week of waiting. And within this week, the patient already is admitted to the hospital again to receive some mild chemotherapy. So it doesn't work without any chemotherapy, but the chemotherapy that you need to prepare the body for the T-cell therapy is pretty mild compared to standard chemotherapy used for Ewing sarcoma or other cancer treatment. And it takes 3 to 4 days. After that, you infuse this bag of prepared CAR T cells where the CAR, the chimeric receptor, is introduced into the patient's own T cells to enable it to recognize cancer. You transfer it, and then you do nothing but wait how the patient tolerates the treatment and manage potential complications. And typically the patient stays on the ward for another 7 to 10 days. Just to follow the patient up under inpatient conditions. And then there is follow-up, of course, for disease response and also potential late effects, side effects of CAR T-cell therapy. So here, this is illustrated by just a few pictures. This is a bag of CAR T cells, very small bag for a small child. The number of T cells is adjusted to the patient's weight. So for small children, it's really just a very small amount of T cells in suspension. And then it's simply injected via a central line into this patient while the patient is, as you see here, sitting in her hospital bed and typically doing very well during this very short, maybe 15 to 30 minutes procedure. So after this, the T cells are in the bloodstream. They will go to the site of the tumor, recognize the tumor as foreign, and start an immune response. This is how patients think this is working. This is one of the first children we've ever treated with CAR T cells. And after explaining the whole procedure to the child, he painted this picture where he compared it. So this is like the standard chemotherapy, syringes with medicine. And this is the CAR T cell attacking the tumor with the weapon of these effector cells. And tschüss means bye-bye. So after that, there are a couple of cells just cleaning up the mess. And here's from a more scientific view what happens. The first days, nothing really happens. This is when the immune response in the patient's body is initiated, and this doesn't cause much of a stir. So typically the first days are really boring. And we do this CAR T cell therapy, we perform it on just a regular ward, and the first days are typically spent in the playroom and not in the hospital bed because the kid is feeling entirely well. And then typically the child develops a spiking fever around day 7 to day 10. Sometimes it's a little earlier. And the fever is the consequence of the immune response that has been initiated by the T cells. So it's part of the treatment, and it's a side effect, yes, of course, but it's also part of the immune response. And we are not too unhappy to see it because it really helps us witness the response that is going on. Of course, we give antipyretics, we do what we can for the patient to feel well, and we also try to mitigate it in a way that the patient doesn't have any serious inflammatory reaction. So what happens? The T cells recognizes the tumor cell, and then the T cell releases a lot of agents into the bloodstream that attract additional immune effector cells, because the T cells will need help from other players of the immune response to really eliminate all the tumor cells. And this is what causes the inflammatory reaction, the stir, the fever, and can also cause further side effects. This is an example just from our ward. So again, day 4, day 5, morning of day 6, nothing happening. And then suddenly there's this febrile reaction. At the same time, this is the blood pressure. You can see that the blood pressure is going down, not in a dangerous way here. 54 is the lowest. It's still okay for this child here. So we were never really worried. But when this starts, when the blood pressure is affected, there's always fear that the immune system might cause the patient to go to the ICU because some blood pressure support might be needed. And at that point, we always give an agent that counteracts this overshooting immune response. And this typically helps to prevent any stay on the ICU. So giving fluids, giving antipyretics, giving this agent in the hands of an experienced team typically can avoid life-threatening complications in our patients. And then the temperature goes down again and the patient feels fine again. So compared to what these patients have gone through previously, typically they tell us that they were really surprised how well it all went. But there's another potential side effect, that's neurotoxicity. And neurotoxicity, of course, always is very scary. This means that these agents that have been released by the T cells and by the other effector cells of the immune system have traveled to the brain, and that can cause a child to go to sleep and really sleep for long periods so that you can really get worried, or it can cause seizures. It can cause a tremor, so patients tremble, cannot really write properly anymore, and can have all kinds of neurological symptoms. They're reversible in the case of these leukemia CAR T cells. I've never heard of a neurotoxicity that was irreversible in the patients treated with the standard CAR T cell agent, but it can still be pretty scary. So we had patients who really went to sleep for 24 hours and had to go to the ICU to be in a secure environment. And then we gave several drugs, steroids in this case, and also this specific agent with which you counteract the immune reaction. And then the child woke up again and was completely fine as previously. We also had children who had seizures, so we are giving anti-seizure medication now in advance. And what we do, and this is what is illustrated on this slide, we always do, it's in German, but it's still pretty easy to understand. So we have this sheet for every age group. And so we let the children tell us, so what do you see here? And sometimes the child really looks perfectly normal, but is unable to tell us that this is an apple. And at an age where the child really should be able to do it. And an early sign of neurotoxicity often is that they can no longer really follow this line with a pen. In adults, you would ask them to write their name or to write a little message. But in children, we just do it here. And at older ages, we let them show us to more complicated objects. So we have this for various age groups, and it's a very simple test that we do twice a day. And it really helps us assess the first signs of any neurological reaction. So I know you don't want to hear about leukemia, but I really had to introduce the CAR T cells, and you need leukemia to introduce them. Actually, one of the first in human and first in child trials performed with CAR T cells was not performed in leukemia. Even before leukemias, we had developed CAR T cells for another childhood cancer, neuroblastoma. And the reason why I started with leukemia is that in leukemia, the true success story has evolved, whereas in solid cancers, CAR T cells are not yet really effective. And this was also the case in this first study. Who you see here is Malcolm Brenner. He is the director, or was at the time the director of the Center for Cell and Gene Therapy at Baylor College of Medicine in Houston. And I was a postdoc with him now 20 years ago, more than 20 years ago. And we together had read this paper from Zelig Eshar and had this idea of developing CAR T cells against a marker of neuroblastoma cells, because this is what you need to make CAR T-cell therapy work. You do need a surface-expressed marker that distinguishes your cancer cell from a normal cell. And neuroblastoma is characterized by high-level surface expression of something called GD2. And you see it in this histology slide. So everything green is GD2, and it's expressed reliably on the surface of all the tumor cells. So we made a CAR against GD2 and made CAR T cells and started a clinical trial. And we did see responses in a few patients, but these responses were really only moderate and they were temporary. So we were not able to save any individual child with our GD2 specific CAR T cells. So that was the state 20 years ago. In the meantime, other centers have developed GD2 specific CAR T cells. There is a pretty promising study, not yet published. That's why I cannot show you any examples, in Rome by Franco Locatelli. And there's a published study here by a clinical research group at University College of London. And they have found some evidence that CAR T cells can be active against solid cancers. This is again not Ewing sarcoma, it's neuroblastoma. And this child was not cured, but the child had a very impressive response. So what you see here is bone marrow, and all the dark red or dark or magenta spots are neuroblastoma cells. And they were all removed here by CAR T cells on day +29, except for this little island of cells still present. So it was not a complete response, but a near complete response. And it really shows the power of CAR T cells. And this is imaging, not very easy to see the differences. But what I would like to draw your attention to is the vertebral column. These are all metastases from in a neuroblastoma. It's a different neuroblastoma patient treated with the same type of therapy. And these vertebral column metastases were not completely, but nearly completely eliminated by the CAR T cells. So there's hope on the horizon, even though in this study, none of the patients was cured with the CAR T cells. And one further example from the United States, in Stanford, there was a study where children with a fatal brain tumor received GD2 specific CAR T cells that were injected either into the peripheral blood or directly into the brain. And you see here some images from one of the patients where after one dose of CAR T cells, the tumor was at least getting a little bit smaller, and then the tumor came back and the patient received another dose of CAR T cells, this time given directly into the brain. And again, there was some response. Again, no cure, but that's how clinical research starts, with at least some advance. And this is a clinical picture of a child who was not able to really move her fist anymore. And she was now able to extend her fingers again after CAR T cell therapy. So there is something that these GD2 specific T cells can achieve. And now I'm finally moving to Ewing sarcoma. We have looked for adequate antigens in Ewing sarcoma for a while. And many other groups also have tried to identify antigens that can be targeted by CAR T cells in Ewing sarcomas. There has been a lot of frustration there. There doesn't seem to be a surface antigen that can reliably distinguish Ewing sarcoma cells from normal cells. There are a few examples now that can be targeted by CAR T cells, so it's not just GD2. But we have found that GD2 can be quite an adequate antigen in Ewing sarcoma. So we have looked at now even a lot more than 100 Ewing sarcoma biopsies. And we have found that this antigen described in neuroblastoma can be expressed also in a high proportion of Ewing sarcoma cells. 2/3 here, and it was consistent at diagnosis and relapse. But what we found is that GD2 expression in its density can be variable among patients and also be heterogeneous in individual tumors. So here's a situation where we think GD2 specific T cell therapy could be promising because every tumor cell expresses GD2. But here you see another patient where GD2 expression is on, you must believe me here, but pathologists would confirm it's expressed on every cell, yes, but it's only moderate to mild intensity, and the surface expression might not be strong enough for the CAR T cells to detect GD2. And here you see a situation that we see quite often, that a proportion of Ewing sarcoma cells are GD2 positive, but other cells are GD2 negative. And what will happen here is illustrated in this graph up here. These tumor cells with lack of GD2 or low expression will be able to hide from the CAR T cells. So you will eliminate all the GD2 positive cells, but the tumor can still grow back. So we think at the moment that only patients with high and homogeneous surface expression of GD2 will qualify for GD2 CAR T cell therapy. And we expect that to be the case in around maybe 30%, 20 to 30% of Ewing sarcoma cells. So at the first step towards GD2 CAR T cell therapy, or in every trial, we will have to check for the GD2 expression status. And if that is not high or homogeneous, then the patient unfortunately will not be a candidate. So we will have to come up with additional CAR T cells against alternative targets or against more than a single target, but at least it's a start. So, but even in a tumor that has consistent GD2 expression on every single cell, even in those tumors like neuroblastoma, the tumor eventually grew back and no patient really was cured with CAR T cell therapy alone. The reason for that is that these tumors have barriers in their tumor microenvironment. I showed you this image initially to illustrate that tumors contain a lot of other cells than just tumor cells, for example, some T cells, but they also contain cells that protect the tumor cells from T cells and that help them grow uninhibited. And especially Ewing sarcoma has a lot of immune cells that are not exerting any immune function, but is protecting Ewing sarcoma. And very likely, this is the mechanism how Ewing sarcoma, neuroblastoma, all these tumors are protected against also adoptive transfer of CAR T cells. So we will have to do something against this tumor microenvironment. We have to strengthen the CAR T cells to overcome the microenvironment. And that is the concept of the study we are planning. We are engineering patients' T cells with a CAR against GD2 because we don't have any beta target at the moment. In addition, we put in another gene that will result in release of an enhancing agent as soon as the CAR T cell meets its target. So when the CAR T cell engages GD2 on the surface of a Ewing sarcoma cell, this will result in elimination of the cell. But at the same time, the T cell will release this agent, and this agent will go into the tumor microenvironment and help get rid of the hostile cells that protect against T cells. And this agent will also act back on the CAR T cells and make them more potent within the hostile microenvironment. So that's the theory, and it's also the science behind it, because of course we have done a lot of preclinical research that makes us hypothesize that this is the mechanism of action that will be that can act also in vivo in the human patient, but we haven't really tested this in humans yet. So this is all, the point where you have to start a clinical study, and that is what we are planning to do. The study is a study in Germany that is funded by the Ministry of Education and Research. Such clinical research is extremely expensive. So it's several millions of euros or dollars you need to perform such a clinical trial, because CAR T cells are very expensive to be manufactured. So it's even more expensive than a clinical study with a standard agent. You need a preclinical team that designs the vector and does the preclinical research also in in cell cultures and also in animals and mice to show that this is really promising enough to for a clinical study to start. We need experts on cell manufacturing because each individual product will have to be manufactured in a lab under pharmaceutical safety standards, because of course there shouldn't be any unnecessary risk for the patients. And I'm coordinating the study together with an adult oncologist because we will enroll not only pediatric patients, but also adult patients. And the major prerequisite is that they have a GD2 positive solid cancer, and we will test that initially. So this is how it works. Patients with neuroblastoma, Ewing sarcoma, osteosarcoma, and even breast cancer, another cancer that can have GD2 expressed on the tumor cells, will qualify for GD2 testing. We do that in a centralized lab, according, of course, to a standardized, validated procedure, so that no patient with insufficient GD2 expression is exposed to the risk of study treatment with no perspective of potential benefit. If we consider a patient GD2 positive to sufficient extent to be eligible for the study, the patient will go through the enrollment procedure. There are inclusion and exclusion criteria like in every clinical study. And then we will perform a dose finding part. That is what we always do for something as new as this. So this is really first in human, especially with the IL-18 enhancer. In the context of GD2 targeting, it's a first in human product. So we have to be very careful and start with a careful dose and then go up to a dose that we consider sufficient to see benefit. And we will do dose finding independent in the pediatric and adult population because of course the dose in a child could differ substantially from an adult. After that, we will select the most adequate dose, and then we will extend our individual disease groups so that we can have a first preliminary assessment of potential benefit across the individual disease groups. And it's possible that in neuroblastoma, we could see some signs of activity, whereas in sarcoma, it will not work, or it will work in breast cancer, but not in the other entities. And that's why we do this in separate cohorts. So that's the essential outline of the clinical study. It will be performed across Germany with 2 sites manufacturing the product, and overall 4 cities serving as either pediatric or adult trial sites or both. And the product will be shipped to the sites that don't have manufacturing at their own site. So that's a decentralized academic manufacturing, which is something that we think will be needed for rare cancers treated with innovative cell products. We will initially not be able to treat any patients outside Germany, but after the first safety part of the study, it may be possible to accept international patients. And we have no idea at the moment how competitive this will be, but the idea is to have at first a multi-center but national trial. So just a few words concerning this academic manufacturing and decentralized supply. That is also something that is very important in the context of this new type of treatment. This is not off the shelf, so it's not a chemical agent that can be produced and upscaled to treat thousands of patients. This is something that needs to be prepared in a patient individual manner, even in the case when the agent has been has shown benefit, is approved. So in order to offer this type of treatment to patients with a rare cancer, we will have to find a new way of manufacturing, because it will be very difficult to find a company that wants to further develop this very expensive, for a company it's very expensive to develop, product and then sell it to sites for even a high price will not reimburse for the research and development process. It will be very difficult really to advance this in case it shows efficacy. And so the academic community at the moment is really busy to find concepts how this could be distributed. And in Europe, we are currently working on a decentralized platform where at least one pediatric oncology center per country is able to manufacture such products so that it can be transferred to trial sites within that country. So an individual product, but still according to standardized procedures, can be manufactured, for example, in Italy, in Rome in Italy, in Paris in France, in Münster in Germany, within one clinical study that treats patients all across Europe. That's our vision. And we think this will be necessary. And I know my colleagues in the United States are also thinking about such approaches at the moment. So where we will go in the future, I do hope that CAR T cells will make an impact also in Ewing sarcoma. I am realistic enough not to think that our study alone can really change the field towards GD2 therapy for everyone and no more chemotherapy, whatever, but we still hope that it will make a difference. The major impact that T-cell therapies could make is that they could result in plateaus of relapse-free survival. And that is because T-cells have this property of staying behind in the body and having some memory function. And that is what we see in the leukemia patients, that in the long-term survivors, the CAR T cells stay behind and protect against further tumor re-challenges. And so that's what the achievement of T-cell therapy could be, but very likely we will have to combine it with standard multimodal agents, probably also with molecularly targeted therapies to attack the tumor from various individual sites. I don't believe that CAR T-cell therapy alone will be able to cure a majority of Ewing sarcoma patients. But it may become a certain component of Ewing sarcoma therapy in the future, or it may not, but I'm doing what I can together with my colleagues to advance it. And so this, again, an image or a very nice picture I find drawn by a patient with a pediatric cancer. And I think it's the most beautiful picture I've ever seen of a CAR T cell. So with this, I'm done and I'm happy to take any questions you may have. And I see that there are a couple of questions already addressed in the question and answer forum.
Great, thank you so much. You are a great teacher on top of a brilliant doctor and scientist. So thank you so much for breaking that down for us so clearly. We do have a lot of questions that have come in. First, when we were looking at that chart of cold and hot tumor environments, does Ewing sarcoma typically fall into one of those buckets, or is it really tumor, child or tumor dependent?
No, Ewing sarcoma is one of these cold tumors. Oh. No or almost no T cells infiltrated. So it's really black and white here. And it's black. Yeah.
I guess a follow-up question to that, in a tumor biopsy, can you detect whether it's cold or hot, or can you detect that with conventional testing, or is that something that's very specific?
Yes, not necessarily on a standard biopsy, but there are ways now to stain for the individual populations that are present in a tumor tissue. And we've done that, and that's how we know that Ewing sarcoma is a cold tumor. We have actually, it's typical science. We have searched for T cells in Ewing sarcomas and also for this marker that can be blocked by checkpoint inhibitors, because we were still hoping at the time to be able to successfully use checkpoint inhibitors in Ewing sarcoma. In the first biopsy we looked into, we did find both T cells and this checkpoint. And then we looked at 100 further samples and never found it again. So this was an exception. And that also really tells you that in science, you should never really trust a single finding. And that's what happened to a lot of my colleagues. So there are rare exceptions, but even these exceptional patients that have some T cells present do not respond to checkpoint inhibitors.
Okay. Thank you for that.
I think, staying on that topic of biopsies, is that how GD2 or the sufficient level of GD2 is detected? And would that be something seen on standard sequencing? Or is there some advanced sequencing that needs to be conducted?
It's standard staining of tumor tissue. It's not sequencing. In fact, I didn't say that. I didn't mention that this GD2 is a sugar. And what you can detect by sequencing is proteins. And GD2 is not a protein. It's kind of a sugar lipid component. And it's completely missed in all these sophisticated sequencing screens. But we have a very reliable technique to look at tumor tissues and detect it in tumor tissues. And it's a tricky technique, which is one reason why we have to do it in one standardized lab. But we can very reliably detect it there. We would not ask for a new biopsy. We would also use a biopsy from a former tumor manifestation. For the, typically the relapse is diagnosed via a biopsy, and that's the biopsy that we would use. But if the patient then has further progressive disease and we would qualify for a phase 1 study, we would not necessarily ask for new tumor tissue, but it's always better to look at the current status, of course. So if you could easily do a rebiopsy, it could be a good choice, but we would also accept tissue from an earlier time point.
Would patients from overseas be able to send in samples for you guys to verify the GD2 level expression? Is that something that would be possible, or they need to be in Germany to do that?
No, I mean, I don't know how large the amount of samples would be. And at the moment, I would really be hesitant to encourage too much inclusion into this trial. Sometimes these trials really are started and are stopped again after one patient because the treatment is toxic. It can still be toxic. GD2 specific CAR T-cell therapy has not been toxic in the clinical studies in Stanford, in Rome, in London. But we are introducing a new innovation here, and that is this IL-18 and this enhancer. And the enhancer could change the picture completely. So it's possible that we have to go back to a lower dose level. And this is really nothing. I mean, I would love to encourage everyone to become a candidate for this study, but it's still too early at the moment. So in general, I wouldn't exclude staining tissues, and it's not very time consuming. And we are sometimes doing it for patients within Europe or within Germany already. But I think it's too early to look at. I would really, by the time we can, if this really flies and gives us hope, I would do everything to extend it to other sites. And I do have very strong collaborators also in the United States. We would do what we can to include patients. But at this moment, it's still too far from a new chance for a Ewing sarcoma patient to stain tissues and then wait for the study to open.
Right. Thanks for bringing up IL-18. That seems to be like a key point of differentiation on your clinical trial based on anything else that's been done in the past. Would you say it's fair to say that really the novelty of this trial is GD2 plus the IL-18 relative to anything else that's been done for solid tumors? Is that a fair statement to say?
Yes, definitely. I think that this enhancer is our chance to break up the microenvironment and to enhance the power of the CAR T cells. And it's the theory behind it. It's the hypothesis we're testing, and it does have potential, but mouse experiments don't really go all the way. And I've seen in my scientific and clinical life, I've seen a lot of very promising mouse experiments and then a very disappointing clinical study afterwards. And there could be two disappointments. One is it doesn't do what it promises. So the T cells are still too weak, and they still are not really active against the tumor. And we do see some responses, but nothing durable. That's one fear. And the other fear is that it becomes toxic by this enhancer. And that because GD2 is also expressed on some normal cells in the body, on cells in the brain, for example. And I still think it's very brave of Robbie Masner in Stanford to treat patients with a brain tumor, even with intra-brain T cells, against a target that is expressed at low levels in the brain. I was so scared that these patients would endure terrible CNS toxicities, which really, even in a child with an incurable cancer, would be a devastating side effect or complication to observe. And I don't want to be the doctor injecting a T cell product that causes such devastating toxicity. But he did it and nothing happened. It was fine. And so for some reason, these CNS cells with low GD2 expression seem to be protected against CAR T cells. But we cannot be entirely sure before performing this phase 1 dose escalation that these CAR T cells will remain safe with IL-18. And we have of course, we've done a lot of preliminary assessments in everything we can do to protect study patients from something potentially very toxic. And what we do now is that we start with very careful doses. But it could be that we start seeing neurotoxicities very early on, or some completely unexpected. With first-in-human studies, you can observe something completely unexpected that could make us, cause us pull back with this whole approach again. But that's part of the development process, unfortunately.
Can I just follow up on IL-18? How did you guys determine that as like an ideal partner? Is it synergized with the GD2 targeting, or is it more overcoming the immunosuppressive tumor microenvironment, or both?
Yeah, we know just by studying the individual components of the immune system, like from the scientific point of view, we thought about what could be rational candidates that fulfill this purpose. And we came across IL-18 and thought it's a wonderful candidate for several reasons. It does something positive to the tumor microenvironment. So it seems to turn off our enemies and support the cells that help the CAR T cells. It does have an effect on the T cells themselves to become more potent, more powerful in difficult environments. It also is safe in humans. If you inject IL-18 into the vein, it causes some inflammatory reaction, but not severe. So you seem to be getting away with some IL-18 also released into the system. And of course, a lot of the IL-18 will be deposited into the tumor microenvironment because the T cells only make it when they engage target, but still something will go into the blood. And it's not toxic on itself. So that made us do experiments, and these experiments, first in vitro and then in animals, pretty much supported the idea. So the CAR T cells supported with IL-18 are much more potent in the mouse model than cells not supported by IL-18, but a mouse model is a mouse model, but nothing more. It doesn't necessarily represent the situation in patients. And there's nothing we can do to approach the situation even more closely. We have to go into a clinical study now and protect the patients, the study patients, by starting with very low doses. That's all we can do now. But so far, it looks like our hypothesis is confirmed, and we will have to see.
Thank you.
Will you be accepting relapsed patients into your trial, or is this primarily for newly diagnosed candidates?
No, the patient is restricted to patients with incurable situations, relapsed or refractory after or to standard treatments. So as you always have to do with something as new as this and potentially still toxic, causing something unexpected, you always have to start with a population that really has exhausted all established treatment modalities.
Can you touch on
that it becomes part of standard treatment in the future, because we don't want to treat patients with refractory disease, because we want to treat patients in a curative manner from the start. That is the ultimate goal. but we're not there yet. We are far from there.
Cool. Thanks for that, Dr. Rössig. Can you touch on some of the inclusion criteria, age, et cetera, some of the basics?
Yeah, children and adults, children even from the age of 1 year, so only first year of life is excluded. And then the patient has to be in an acceptable condition still, with some life expectancy of a couple of months. So it's a typical phase 1 population, so nothing outrageous than the heart function, the kidney function, and so on. I think that's all criteria that many patients will fulfill. The difficult entry criterion really is the GD2 expression status that will exclude a lot of Ewing sarcoma patients. Not so many neuroblastoma patients, but Ewing sarcoma patients.
But in this trial, if I'm understanding, you are kind of reserving spaces for Ewing sarcoma patients, that if, you know, there's a high number of qualified breast or osteo, still make sure to try to get those.
For the dose finding, it's not really so relevant what kind of disease. So we will first comers will be treated, and that also only distinguishes between pediatric and adult patients. So we will have to fill the dose toxicity levels for all for both age groups. But after that, we have made sure that we include a sufficient number of Ewing sarcoma patients to come to a first conclusion whether this is any promising at all or not. And that is made sure, yes.
There's some questions on here with regards to other CAR T therapies in the US, specifically at St. Jude's and Seattle. Do you know if those trials are looking at GD2 or the other biomarkers? I know there's one on B7H3.
Oh, yes. There are several GD2 specific CAR T cell trials ongoing, and all these people have realized that a GD2 CAR T cell alone, like an old-fashioned GD2 CAR T cell alone, will not do the trick, and that you have to support it with something. And they're all thinking of something, and there are several studies ongoing where some advance is already in there. Not IL-18, but something else. We don't know which one will work in the end. It could be IL-18 and something else. It could be, the winner could be something completely different. We don't know, but it's certainly no, I mean, I feel honored that you're asking me to give this talk, and we are happy that we are in Europe, we are contributing to the field. But in fact, the CAR T cell field is more advanced in the United States, because a lot of these hurdles of manufacturing are even more difficult to be addressed, regulatory or hurdles more difficult to be addressed in Germany and in Europe in overall. So definitely, so if you are looking for treatments, look at Stanford and all St. Jude's, Baylor, so Houston, Texas Children's, and these institutions. UMC Leinefelter, there are several institutions. So, and you find it when you just look for CAR T cell trials, you will find all these institutions. And many of them are now addressing GD2. There are some other targets also in Ewing sarcomas that I didn't have sufficient time to look at. And we've decided to go for GD2 as the first run.
Okay.
All right. Well, we promised we would let you finish right at 6 p.m. your time. We appreciate you taking the evening. I just have one more question. The Ewing's population by necessity is very hopeful, and this is really exciting therapy. You know, if all goes according to plan for you, what is the kind of the rough timeline of when you could see a phase 2 trial begin?
This study is scheduled for a duration of 2 years. Which is always too optimistic. I know that, and it always has to do with funding. That the funding sources always ask you to finish the study in 2 years. We may take a little longer than that, but by starting the phase 1 study in mid-2023, we expect to be done by end 2025. So that would be the start of phase 2. But by then we will already at least get the basic questions answered. Is it too toxic to be pursued? or not? And if not, are there signals of efficacy? And that's the good thing about CAR T cells. You typically get the answer. Is there any promise in it or not at all? You get it pretty quickly. So in case of a positive development, it will be 2025. In case of a negative development, we may know earlier. But of course, we hope for the first.
It's going to be
Thank
great.
you for thank you for that, Dr. Rössig.
Yeah.
This has been fascinating webinar series. We'll be sure to post it. Thank you for your time. If you need to sign off, absolutely right now, we'll wrap things up with the community. But thank you for being here, and thank you for giving us hope as patient community. It's very much needed, as you know, and it's not an
Thank you for the
easy
honor
work,
of letting me speak about my favorite topic. And
Absolutely.
yeah, thank you all in the audience for listening. Bye-bye.
All right, have a good evening. For the rest of you that are sticking with us, thank you to our viewers for joining us today as well. We have several viewers across the pond, which is always exciting for us to see. And thanks for those of you that took our survey after our last webinar series. We heard you guys loud and clear. We will indeed be making this a monthly webinar series. And on that note, we have a follow-up discussion in February 16th, specifically at 2 p.m. Eastern. We'll be discussing an important but somewhat controversial topic of stem cell transplant for relapse disease with Dr. Dave Margolis from Children's Hospital, Wisconsin. This is a topic that comes up over and over again on the Ewing discussion boards, and hopefully it will lead to some very healthy and insightful discussion amongst attendees and Dr. Margolis. So mark your calendars, February 16th, 2 p.m. Eastern. And as always, please be sure to subscribe to our Ewings U emails. You can do that by going to littlewarrior.org, navigating to the Ewings U tab on our website, and also follow us on Instagram and Facebook, as we always post about our next speaker that way as well. Lastly, in closing, we have a sign off here at Little Warrior that embodies our fight in honor and in memory of so many little and big warriors who have faced this cancer. It's how we sign off on all our communications, both internally and externally. So wishing you all a healthy and happy new year in 2023. On a count of 3, we're gonna do a swords up. 1, 2, 3. Swords up, everybody. See you next time. Thanks for joining us.
Great day, everyone.